A very good morning to all of you. Once again, welcome to the teaching class of Department of Urology, Savai Mansingh Medical College, Jaipur. Today, we have a presentation on tissue engineering. So this would be presented by our resident, Dr. ASHOK Rajpura, and would be moderated by our faculty, Dr. R.D. SAHU. So I would request Dr. Rajpura to kindly share his presentation.
Thank you, sir. Good morning. My topic is tissue engineering. Introduction to tissue engineering. Tissue engineering combines biology, material science, and engineering to develop functional replacement for damaged or diseased tissue. Key component of tissue engineering: cell, somatic cell, stem cell, scaffold, biodegradable polymer, decellularized tissue, signaling molecules, growth factor like vascular endothelial growth factor, tissue growth factor, beta. Bioreactors provide control environment for tissue maturation. Cell use: somatic cell have limited proliferative ability, and stem cells types: embryonic stem cell, induced pluripotent stem cell, somatic stem cell, and perinatal stem cell or fetal stem cells. Somatic cells: fully differentiated specialized cell obtained from normal tissue. Example: cultured epithelial cell grown in vitro and used to treat the patient with burn injuries. Tissue taken from autologous allogenic cells. Autologous not required immunosuppressant. Limited proliferative ability in vitro. Stem cells undifferentiated or non-specialized cell through cell division to renew themselves independently. Classification: embryonic stem cell from early embryo, fetal stem cell from fetus, adult or somatic stem cell from normal tissue, induced pluripotent stem cell from reprogramming adult specialized cells. Stem cell types: totipotent, pluripotent, and multipotent. Totipotent is the ability of a single cell to divide and produce all the differentiated cell in organism, including extraembryonic tissue. Pluripotency is capability of differentiating from to all the three layers, germ layers. Multipotency is the ability of a stem cell to produce only a closely related family of cells. Totipotent stem cell from zygote that is capable of differentiate to germ cell as well as extra embryonic tissue, and a pluripotent cell taken from inner cell of blastocyst and differentiated in three germ cells. Multipotent stem cell taken from adult tissue and differentiated to relative family of cells. Somatic stem cells capability to differentiate it into a limited number of specialized cells. Multipotent example: replace sentient or damaged cell in the blood and gut mucosa. Types: hemopoietic stem cells, treatment for hematological malignancy. Mesenchymal stem cell used for tissue engineering in endothelial, progenitor or stromal cells, and neural stem cells. Mesenchymal stem cells source is bone marrow, adipose tissue, and umbilical cord. Morphological resemble fibroblast differentiated into mesoderm mainly and ectoderm and endoderm. Isolated from iliac crust. Aspiration, liposuction used in burn patient and repair of cartilage defect. Embryonic stem cells is totipotent, obtained from inner cell mass of early human blastocyst. 4 to 5 days of fertilization. Shows circulant embryos created through IVF for treatment of infertility. A recipient somatic nucleus injected in the homologous embryonic stem cell can be created. Disadvantage is allogenic and risk of rejection is there. Ethical limitation and political debate. Teratoma. Inducible pluripotent stem cell is retroviral or lentiviral transfection to introduce transcription factors. Is pluripotent and autologous. Problem is by using retrovirus genomic integrative virus may lead to activation of oncogenes. To reduce this risk, non-retroviral vector can be used. Example, adenovirus and Shanghai Advantage is no rejection and no ethical issue. Disadvantage is procedure may take several weeks. Fetal stem cells source is aborted fetus is pluripotent. Use is in diabetes, Parkinson disease, spinal cord injury. Clinical benefit is controversial, ethical and political issues there. In vitro differentiation of stem cell to specialized cells, addition of certain chemicals, cytokine growth factor H at specific point during culture. Example is exposure of iPSC to retinoic acid and BMP4 promote keratinocyte formation. Culture of iPSC on matrigel scaffold with ascorbic acid exposed to GSK inhibitor and anti inhibitor promote immature cardiomyocyte development. It is essential to verify purity of a differentiate cell and confirm the absence of a undifferentiated cell to reduce risk of tumor transformation. Scaffold. Anatomical arrangement is absolutely necessary for normal functioning. Stem cell grown in vitro are unable to spontaneously achieve desired organ shape. Hence, appropriate scaffold must be necessary to give shape. Example 3D rigid and semi-rigid porous scaffolds. Source natural source is intact human tissues, small intestine, submucosa and bladder mucosa, artificial implantable biomaterial, polyglycolic acid and polyglycolactide. Requirement of scaffolds provide structural support for cells, allow cell to attach, migrate and proliferate, enable oxygen, nutrient and regulatory factor access to all cells, deliver signal to promote cell migration and proliferation. Technology used for scaffolds, computational designs, 3D printers and electrospinning, cell shading of scaffolds, static cell shading, dynamic cell shading, magnetic cell shading, vacuum shading system and scaffold perfusion system using bioreactors. A challenge to tissue engineering, delivery of oxygen, delivery of nutrients, plate tissue growth easily, tubular structure difficult to grow. Example, blood vessels and colon, solid organs, greatest challenge, example, kidney. Safety concern regarding tissue engineering is malignant transformation, infection, graft rejection, need for immunosuppressive therapy. This is a example of porous scaffold in research development. A is a macroscopic view of collagen scaffold and B is a microscopic view show the structure of collagen scaffold. Cells working with other population and differentiated into multiple lineage, osteoblasts, chondrocyte, adipocyte and other. Application in urology in bladder reconstruction, engineering bladder using autologous cell, urethral repair, acellular matrix and tissue engineering graft, kidney tissue engineering, bioartificial kidney, nephron units used in ESRD, penile reconstruction, bioengineering, corpora cavernosa, urinary incontinence, stems, cell repair, base repair, sphincter, erectile dysfunction in Peyronie disease and in infertility. Tissue engineering in urology mainly used in diagram showing bladder repair, penile reconstruction, urethral defect and kidney. Tissue engineering of urethra either using congenital defect or post-traumatic defect. Urethra either use engineering tissue use either as page graft or tubular graft for replacement of wall of circumference urethral segment. Page graft used for partial urethral defect in case of poor vascularity, unhealthy bed, and long urethral defect. Naturally derived collagen-based material such as woven meson of polyglycolic acid without cell and with cells, bladder-derived acellular mucosa, acellular urethral submucosa, and collagen gels. This is a study conducted by Rava and Rivera et al. In 2011. 5 patients with urethral injury included in study had a small tissue biopsy taken from a cell and cultured in vitro seated in two layer on tubular scaffold that were implanted surgically. Engineer urethra were able to show adequate anatomy both by urethroscopy and urethrophy and function long term. Age above intra-photo showing tubularized graft implanting in urethral defect. 5% included in study. First line showing patient uh, before surgery, and the second line showing in all patient 12 months after surgery, and at last follow-up, third line showing complete defect is covered by tissue engineer graft. Urinary bladder reconstruction. Regenerated bladder should a compliant muscular wall, well differentiated urothelium. Acellular and cellular approach can be used. In acellular approach, scaffold recruit cell for new tissue formation, commonly used. Result, non-sedated scaffold fail to show full generation of the bladder wall. Reason for failure is early exposure of scaffold and newly implanted cell to urine, extensive scarring with the graft. In cellular approach, cell taken either from bladder mucosa or intestinal submucosa, urothelial cell culture in vitro, and sedated on biographic scaffolds, then cell culture in bioreactors, grafted and engineered tissue replaced in bladder. Bladder cell therapies, injectable therapy used in stationary incontinence and vaginal reflex, use either autologous smooth muscle cell or myoblast. Recent approaches are directed to finding new source of a cell for bladder regeneration. It is hypothesized that both epithelial and dermal multipotent stem cell population within hair follicles should raise new possibility for tissue engineering of urinary bladder. A study conducted in 1998 showing reconstruction of a urinary bladder by Atala et al. And a pilot study of 7 patients were reported by Atala et al. In 2006. Patient underwent reconstruction with engineered bladder and covered with omental coverage. So increase compliance and decrease and filling pressure, increase capacity and longer dry period or time. Figure A is showing scaffold material shaded with shell for use in blood repair. B is shaded scaffold is anastomosis to natural bladder with Vicryl 40 in the running suture. C is showing implanted covered with fibrin glue and momentum. In ureter, various animal studies, non-sedated acellular graft, cell-sedated biodegradable polymer scaffold, and hybrid scaffold. A study conducted by Osman et al. Investigation the replacement of 3 centimeter segment defect by a tube of acellular matrix to same length and width derived from canine ureter after decellularization technique. Male genital and reproductive tissue uses, erectile dysfunction, period disease, penile cell therapy, reconstruction of corporal smooth muscle, and engineering penile prosthesis. In testis, sliding cell by using testicular dysfunction or anarchic approach is transplanting leading cell or testicular prosthesis. Renal structure mainly using ESRD, either by nephron unit or artificial kidney. One of the most difficult tissue to replicate in the laboratory. Application of regenerative medicine and cell therapy and bioengineering with ECM scaffolds. Treatment of peyronie disease. Successful results were reported in regards to using cellular or acellular tissue engineering scaffold in the treatment of tunica albuginea defect. Shocker studies show that acellular matrix graft of tunica albuginea could to cover a 3 centimeter tunica defect in 9 dogs. All dog develop a straight rigid erection beside colonoscopy showed patent corpora cornosa in all animals. These results suggested the feasibility of tunica albuginea reconstruction in conditions such as trauma or Peyronie disease, treatment of erectile dysfunction, use as replacement of a damaged corporal body, surgical penile reconstruction in conditions such as ambiguous genitalia and extrophy epispadiasis complex is a challenge because of the limited amount of native cavernosal tissue available. Having additional autologous corporal tissue might be beneficial to patient undergoing such surgery. By using 3D acellular collagen matrix, Folke studies reported that a feasibility of developing corporal tissue consisting of human cavernosal smooth muscle and endothelial cell in vivo. This is a diagram showing complete human penile shaft scaffold for composite tissue engineering, organ decellularization and characterization. In day 0 and day 3, day 7 and day 4, penile tissue engineer graft used for either in trauma or transgender surgery where complete loss of penis happen. In use in male infertility, bone marrow stem cells growth to male germ cell was shown in both animals and humans. In addition, if transplanted to the testis of a busulfan treated infertility mouse model, it appeared to differentiate it into germ cell, Sertoli cell, and Redding cell. Advancement in tissue engineering, decellularized scaffolds, 3D bioprinting, microfluidics, gene editing, smart biomaterials. What is 3D bioprinting? 3D bioprinting is a technology where biolink mixes with living cell as printed layer by layer into a natural tissue like the 3D structures. Need for 3D bioprinting. Tissue and organ transplantation fabricated from own body cells. Study tissue and cells to discover drugs alternative to animal testing. This is a diagram showing physical organ model and bioreactive tissue models, 3D printing and 3D bioprinting. Purpose is cell reaction, cell therapy, in situ detection, drug screening, local tissue filling, tissue regeneration, and organ transplantation. In future, personalized medicines, artificial intelligence, artificial organ, stem cell based therapies, clinical trials are future use of tissue engineering. Challenge in tissue engineering is vascularization to ensure blood supply in constructs, immune response to minimize rejection, functional integration, scale up and to decrease the cost of. Thank you.
Thank you, Ashok. So we have, I think, Dr. Bhatt with us, and anybody else who has joined us, if they want to make a comment, they are most welcome. They can just raise your hand or just unmute themselves and let me know, right? And to the residents and faculty also, if you want to ask a question or make a comment on the presentation of Dr. Ashok, please let me know. So in the meantime, I will just summarize the whole thing and then we will take up questions if there are. So tissue engineering, again, applies the principles of engineering and life sciences, and which helps to either refunctionalize, improve, maintain, or restore a body organ, right? So there is another term called TERM, that is tissue engineering and regenerative medicine. So these are few newer terms that have come up with the regenerative medicine. We know about the stem cell therapy. At our center, we did stem cell therapy in muscle-derived stem cell therapy in stress urinary incontinence, which was an ICMR project from the year 2009 to 2015. So 7 years we were into that research, where we developed stem cells in the stem cell research lab, quantified them, characterized them, and then actually with the flow cytometer, we could know exactly the number of cells, and with the various markers, which included CD34, 56, myodin, etc., which will help to know the stem cell property and the muscle property of these cells. So we did the whole preclinical phase of developing stem cells in lab, which was actually proposed to be used for injected in the periurethral region to develop the sphincteric tissue in patients with stress urinary incontinence. So tissue engineering, we know, is basically consists of scaffold, which could be either a natural scaffold or artificial scaffold. Natural scaffold includes various animal tissues also, like cyst. Cyst is the submucosa intestine of the sheep, right? So this has been used as a scaffold, as a graft material in many procedures. And then you have those growth factors, etc., which can be put in these scaffolds, and then cells, either in the form of stem cells or somatic cells, can be seeded into those scaffold, where the growth will help to grow these cells, and the cells will grow into the matrices, ultimately developing into a tissue, right? So this is how the tissue is engineered, either in vitro or in vivo also. So one approach is where you develop the tissue outside the body and then implant it inside. That is one approach. The other approach is scaffold can be used as a carrier for drug or a growth factor. And this scaffold can be implanted in the body where required. And then these growth factors and the scaffold will invite the body cells itself into the scaffold. And these cells will actually grow because of the growth factors which are there, and will actually fill up the matrices of the scaffold. And ultimately, they will develop the tissue, right? And the scaffolds usually either degrade or they may remain as such in the later life. So these are the two approaches of how the tissue is implanted. There are various clinical uses that have been already there. The simplest being use of primary chondrocytes to develop cartilages, right? So this is being used clinically. So primary chondrocytes are cultured and they develop into cartilages, and these cartilages can replace the damaged cartilage in the body. Second, as he said, is the use of skin. So epithelial cells can be actually cultured in vitro, and they can form sheet of skin cells, which can be used to cover complex wounds and burns, right? And then third is to development of bone. And then there is a thing called injector bone, which has come in the market, where the scaffold of these cells can be injected at the trauma site of the where there is an injury of the bone. So when there's a non-union of a fracture, this injector bone can be injected, which helps in the union of bone by regenerating bony tissue. So these are already in the clinical use. Then there have been a lot of human trials also on regenerating bladder tissue, right? So bladder tissue has also been used where it has been engineered outside the body, and then the tissue has been used for augmentation cystoplasty or total cystoplasty. And then there have been uses with urethra for urethral reconstruction. There have been uses for penile reconstruction. Now we are having biodegradable penile implants also. So autologous bone implants are developed which can be used as a penile implant in the body, right? In the penis for erectile dysfunction. And then now we have 3D printers where we can actually print a tissue organ, right? So with the help of bioinks, growth factors, and the 3D printer, we can actually develop tissue like cardiac tissue, kidney tissue, and even penile tissue. Those who have total penile amputation or those who require transgender surgery, the actual phallus can be developed in the by 3D printing system. The basic challenges still are, one is, he said about the immunogenicity. Sometimes it's important. If you're not using autologous stem cells or somatic cells, then there is something where we need to take care of the immune rejection. Secondly, how to vascularize this tissue. That is, again, important kind of a challenge. Third is nerve regeneration in the tissue, how to regenerate nerves so that they actually can have that function of contraction, relaxation, whatever is required. And the fourth, as he said, about the hurdles of ethics, how the ethics play a role, and how the government rules and regulations, again, can become a hurdle or a challenge in the whole this management by tissue engineering. I think this was in brief about tissue engineering, and you may expect in the recent advances of your theory exam a question on tissue engineering or regenerative medicine, no doubt about that. So anybody, president or faculty, want to make a comment or a question? Nachiket, you want to make?
Ladic cell implants, they are very advanced in there.
Right. So again, as I said about male infertility, in male infertility, they can, with the introduction of stem cells, they can actually develop germ cells or help in actually development of germ cells in the testis. They can be developed in vitro also, germ cells, and then can be injected. So there are various techniques which are being used, but still not in clinical application, but research and experiments going on. Anything else? So Dr. Bhatt is there. Dr. Bhatt, would you like to make any comment? And then we have a few more people who have joined us. If you want to make a comment, please unmute yourself. Otherwise, we will go to the MCQs. So question number 1 is, what is the primary advantage of using tissue-engineered bladder constructs over traditional enterocystoplasty? Reduced risk of metabolic complication, enhanced structural rigidity, increased likelihood of infection, reduced cell proliferation.
Reduced risk of metabolic complication.
Yes. So all of you agree? Yes. So yes. So because when you apply intestine on that area, so definitely the function of the intestine, which is compromised, can lead to metabolic complications. So which tissue engineered bladder would not have. Yes. So traditional enterocystoplasty uses intestinality, which can lead to metabolic disturbances due to reabsorption of urine. And tissue-engineered bladder aims to replicate native bladder tissue, avoiding these complications. Which of the following is a commonly used natural scaffold in bladder reconstruction? PCL, PGA, SIS, PLA.
See,
So, as I said, the scaffolds could be either natural, they can be derived from either animal tissue or human tissue. SIS is a very old scaffold that has been used as a graft in so many areas, including Peyronie's disease, etc., for application as a tunica albuginea. So that's the natural, and the rest are the artificial, synthetic graft, which includes PLA, that is polylactic acid, polyglycolic acid, and PCL. So all these are artificial. Is a natural scaffold, biodegradable and biocompatible framework that promotes cell adhesion and proliferation. What is the role of mesenchymal stem cells in bladder tissue engineering? Producing growth factors for angiogenesis, differentiating into urothelial and smooth muscle cells, increasing scaffold biodegradability, and inhibiting immune response.
differentiating into urothelial and submucous.
So, as you know, the stem cells have the property by which various agents, chemicals, neurokines, cytokines, and growth factors, we can actually put them into a certain lineage and make them develop into a particular tissue that we want. So mesenchymal stem cells, which can be derived from bone marrow, it can be derived from adipose tissue, it can be derived from skeletal muscle also. So all these tissues have mesenchymal stem cells, which can be actually differentiated into various other tissues like urothelial and smooth muscles. Which of the following is a current experimental approach in kidney tissue engineering? Decellularized bladder matrix, kidney organoids, and 3D bioprinting, autologous hormone therapy, biodegradable metals.
Kidney organoids and 3D bioprinting.
So the ultimate aim is that the transplantation that we do by taking organs from a donor or from the deceased body, and because we cannot have that number of people to donate, so that to reduce the waiting time and the number of patients which are actually suffering from end-stage renal disease, we are thinking of trying to bioprint the whole organ. And there are a lot of efforts, research going on to bioprint kidney. So we have been able to bioprint few nephrons, which have actually all those components, glomeruli, the tubules, etc., everything. But total kidney tissue, total kidney as a whole, we have not been able to do. People have developed heart tissue, cardiac tissue, and even animals, experiments have been done by putting this heart tissue, cardiac tissue, particularly patients with cardiomyopathy, right? And then we have developed vascular grafts that is already in the clinical practice. So vascular grafts actually bioprinted. So all these have been there, and a lot of research going on, yes. So functional tissue units developed from stem cells and 3D bioprinting is used to create nephron-like structures. Why are autologous cell preferred in tissue engineering for bladder reconstruction? They are more readily available. They eliminate the risk of immune rejection. They provide stronger structural integrity. They have a longer lifespan than synthetic materials. So they eliminate the risk of immune rejection, because we know stem cells could be of various types. It could be autologous, which is the stem cells which are derived from the person himself, right? And the others are embryonic cells, fetal cells, umbilical cord derived stem cells. So those may have kind of immune problem, right? So these autologous cells do not have the immunogenic issues. Which of the following is a novel approach in sphincteric tissue engineering for urinary incontinence? Artificial collagen injections, cell-seeded biodegradable scaffolds, synthetic polymer implants, radiofrequency ablation. So people have used this, but the more important is what I told you about this research that we did by actually developing stem cells in the lab and actually quantifying them and then putting in the periurethral region. So injecting them in the periurethral region of patients with stress urinary incontinence can help to regenerate the external sphincteric tissue, or even bulking them. So it acts as both bulking agent and regeneration of tissue. So stem cells have been used. There have been human studies also. We could not do the human studies because of the drug rules, our DCGA regulation, which didn't permit us. Anyway, so cell-seeded biodegradable scaffolds have also been used for urinary incontinence. In penile tissue engineering, what is the primary target for reconstruction? Urethral lining, corpora cavernosa, tunica albuginea, prostatic urethra. So corpora cavernosal tissue have been tissue engineered in vitro, and they have been used for erectile dysfunction. And as I said, there have been use of penile implants, which have been developed by tissue engineering method, and even the whole penis, phallus, has been developed by tissue engineering, which is still in research, right? Okay, so thank you, everybody. So anybody has any other question? Dr. Bhatt, would you like to make any comment? And anybody else who has joined us? If not, then Dr. Adi. So thank you, everybody. And thanks for joining us today in our class, and have a good day.